In this experiment, we will study the gaseous exchange in green plants in the daytime and at night. Prepare 4 boiling tubes and label them as A, B, C and D respectively. Add 3 cm cube of hydrogen carbonate indicator to each boiling tube. Enclose boiling tubes B and D by aluminium foil. Hang a green leaf inside boiling tubes A and B respectively. Leave 4 boiling tubes in bright light for about an hour. Record the colour change in the hydrogen carbonate indicator in each tube. Compare the results in tube A and tube C. Finally, compare the results in tube B and tube D. In this experiment, boiling tube A represents a condition in the daytime. The leaf in tube A carries out both photosynthesis and respiration. Under bright light, the rate of photosynthesis is higher than that of respiration. There is a net uptake of carbon dioxide. The concentration of carbon dioxide in tube A is lower. As a result, the hydrogen carbonate indicator turns purple. The setup of tube C is similar to that of tube A, except that it does not contain the leaf. It is used as the control. Boiling tube B represents the condition at night. As light is blocked by the aluminium foil, the leaf in tube B does not carry out photosynthesis. It only carries out respiration. Carbon dioxide is given out and its concentration increases. As a result, the hydrogen carbonate indicator turns yellow. The setup of tube D is similar to that of tube B, except that it does not contain the leaf. It is used as the control.